In this video, we're going to be looking at what happens to the products of the endoplasmic reticulum, how those are transported within the cell to the cell membrane and eventually outside of the cell. Now, before we introduce this process, we can probably take this time to do a little bit of a review of how substances are produced using the endoplasmic reticulum. So remember that everything begins with DNA, which fundamentally is the blueprints of how to make every single compound that exists within the cell. But since DNA never leaves the nucleus, DNA produces an intermediate molecule or carrier of the genetic code, which is what we call RNA, and exports that RNA into the rough endoplasmic reticulum. So we know that this is the rough ER because it has ribosomes represented by these little green circles on its surface, and of course the smooth endoplasmic reticulum does not contain these ribosomes. Now, the RNA basically is a set of instructions that is read by a ribosome, and depending on which protein the RNA codes for, it's going to tell the ribosome to produce a specific protein which ultimately is going to be transported out of the cell. Now, in order to do this, we need the aid of yet another organelle in the endomembrane system, which is what we call the Golgi apparatus, although many biologists, including myself, refer to it as the Golgi complex instead. Either one of these names is acceptable. So if we look at the Golgi complex in the context of the endomembrane system, we can think of the DNA as the library of how to make everything, the endoplasmic reticulum as the production center, whereas the Golgi complex we can think of as the transportation or distribution center of the endomembrane system, which is responsible for, on the one hand, packaging anything that is going to leave the cell and exporting anything that is going to leave the cell towards the cell membrane. Although the Golgi complex is actually not just responsible for exporting, it's also responsible for helping stuff that is brought into the cell get where it needs to, and therefore we can say that the Golgi complex also plays a role in importing substances as well. So if we take a look at the Golgi complex, which we can see right in the middle here, we can look at how it fits with the endomembrane system as a whole. So if we take a look at our protein that was produced by the RER, we see that this protein is transported to the SER here because there are no ribosomes, and remember that the SER's job is to produce all of the lipids in the cell, including the phospholipids that make up the cell membrane itself. So we can see that this protein is actually being packaged into a little sphere made of the same material as the cell membrane is, and these are what we call transport vesicles, or simply vesicles. And we can see that this vesicle is transported to the Golgi complex, where the Golgi complex is going to recognize what type of protein this is, and if it recognizes that this is a protein meant for export, that protein would be repackaged into a second vesicle and then carried along the interior of the cell's network towards the cell membrane, which is of course the outside of the cell. Now, because each of these vesicles is made of the same material as the cell membrane is, that means that this vesicle is actually able to attach to the cell membrane, where it would release the protein that ultimately was produced in the RER out of the cell, where the protein would ultimately do whatever it is supposed to do. This protein could be a hormone, meaning that it's meant to send a signal to another cell, or it could be a large structural protein, such as the protein for hair or fingernails, that needs to be assembled outside the cell. But again, the responsibility of the Golgi is ensuring that it gets where it needs to go. Now, technically speaking, the Golgi also is responsible for the reverse process here. So when the cell membrane decides that something is coming into the cell, for example, if the cell is going to absorb a large food molecule, this entire process can happen in reverse, where a piece of the cell membrane surrounds the food molecule and internalizes it into a vesicle, which would then be carried back to the Golgi complex, and then the Golgi complex ships that food molecule to wherever it needs to go. So this gets into the detailed functions of the cell membrane itself with respect to the cell's transportation system. And ultimately, even though the Golgi tells products of the ER where they are supposed to go, ultimately it is the cell membrane that has the final control of what substances can get in or out of the cell safely, and ultimately the cell membrane's job is the prevention and protection of the cell against any harmful substances that want to get into the cell. So ultimately, if we want to think of an analogy for the cell membrane, we can think of it as border security, because not every substance that is found outside the cell is healthy for the cell. For example, if we can consider this bacterium here, and you know that this bacterium is bad because of the angry face that it has here, this bacterium is something that the cell membrane does not want into the cell. But sometimes, just like with border security, the cell membrane makes a mistake and accidentally internalizes something that is harmful to the body or to the cell specifically, such as this bacterium here. So in the event where a cell membrane makes a mistake, what can the endomembrane system of the cell do in order to deal with when the cell membrane makes a mistake? Well, inside specific vesicles that are what we call lysosomes, your lysosomes act as the last line of defense against anything that the cell membrane accidentally brings in that isn't supposed to, because lysosomes are specialized vesicles that actually have digestive enzymes inside, and digestive enzymes are special proteins that are used in order to break down any substance that enters the cell, and this can be something innocent such as food. So in this diagram here, we can see that the lysosome is attaching to a vesicle that actually contains food, and then the digestive enzymes go inside and cut apart this food molecule into small nutrients that can be used by the cell for growth or to produce energy. Now, this also applies to vesicles that contain harmful substances such as bacteria. So if this is a lysosome, the lysosome is then able to attach to this vesicle with the bacteria, release its digestive enzymes into the vesicle containing the bacteria, where they will promptly destroy the structure of this bacterium, killing it and allowing the nutrients that the bacterium is made of to be used by this cell for food or to be able to produce energy. In the next series of videos in this series, we are going to look at the remaining organelles that have more specific functions that are separate from the endomembrane system and ultimately deserve a video of their own.